Right, hello again, grade 11s. We're going to be starting with a new topic, which is animal diversity. This will be I say a lot, we'll just deal with that. The animal diversity topic is very similar to the plant diversity topic in the sense that we'll be looking at different groups of animals and their characteristics. Just to take you back both to plant diversity but actually to classification, when we classify plants, animals, microorganisms, etc., we look at characteristics that make them the same and characteristics that make them different. And if there are enough characteristics, or the characteristics are different enough, then they'll be classified as a group on their own. That is how we came about with plants and animals being in different groups, and microorganisms such as bacteria and protista and fungi being in different groups. So we're going to have a look at animal diversity and the characteristics that each group has in common. Okay, so before I start, you'll see that I'm going to skip through the first couple of slides. The reason for it is this. It is about cladograms or pedigree diagrams, which we also spoke about in plant diversity. But you can't really understand a cladogram if you don't understand or know the characteristics. So I can show you if it was in class, I would have shown you the basic cladogram, made you understand it, then taught you the characteristics and added to it. But I think that with the nature of this type of teaching, I'm rather going to clump things into groups together. So I will ignore the cladogram section and I will start just with characteristics. Hello. Okay, this program that I'm using seems to hang if I've been on a slide too long. So again, I'm just asking for your patience with me. There we go. Right, so okay, so some terms that we'll use just in general, not with any specific group at this point, but just looking at animals. I can divide animals into two groups, but then it would only be two. And those two groups could be vertebrates and invertebrates. Vertebrates being organisms, animals that have a vertebral column, and invertebrates being animals that don't. In other words, they don't have a spinal cord. They don't have a support structure like vertebrates do, like we do. If I talk about morphology, I'm talking about how a structure organism changed its form. And if I talk about sessile and motile, here's the best example. Most of us, I'll include myself, have been very sessile during this lockdown period. We hardly move from place to place. You definitely didn't have to run to my class in order to watch this video, so you were not motile, you were not moving. Okay, if we look at the whole of the animal kingdom, as in all animals together, then they all have certain characteristics. These are the characteristics that made them animals instead of plants or fungi or protists or whatever else. So everyone will have this, and then additional characteristics will make them separated into groups. So first of all, we know that they are all multicellular, made up of many cells. They are heterotrophic. They can't make their own food. They must get their food from an external source. Some still have sex, some still reproduce asexually, and some reproduce sexually. I want you to ignore the embryonic development part as such, because we will talk about it in greater detail. But basically, the embryo will develop into a blastula. So if you remember from grade 9 work, when an egg and a sperm fuse, they fertilize, they're going to form a zygote. The zygote will grow into an embryo, and that embryo will divide into a ball of cells. And that ball of cells, the first time it's divided, is called a blastula. Okay? No animals have cell walls. That's something you also learned in grade 9. Plant cells, cell wall. Animal cells, no cell wall. And animals are all motile. They are able to move from place to place in comparison to plants that are sessile. Animals all have an endocrine system, a system that's going to be responsible for production of hormones, as well as a nervous system responsible for obviously nervous coordination, movement, thinking, etc. And our bodies, animal bodies, are all made up of systems, so reproductive system, digestive system, cardiovascular system, etc. However, these systems may be very, very, very basic in some groups. Okay, right, so starting off then, we're going to talk about body plans. Now, body plans are characteristics, features that these animals will have, the way that they are laid out, that are going to tell you sort of how each organism looks or functions. And we're going to divide it into each of these little topics with a star, each bullet point in any case, so that you understand what each body plan means. Okay, so the first body plan is called symmetry. Now, you should know just from, grade 3 and maths and maths lit and all those funny things, you should know what symmetry is. If you think of yourself as a human being, if I sliced you, if I was some Game of Thrones assassin person thingy, then I slice you between your eyes, through your nose, down your midline, and I'll end up somewhere kind of by your anus, because your legs would divide with that like a chicken. Then you are symmetrical, your left side and your right side. For Mrs. O'Brien's class, I know I haven't taught you, so just deal with me. My class will be rolling their eyes right now. That's okay as well. I know you miss me. Okay, so you are symmetrical human beings. There are different types of symmetry, though, within every group. We'll get to the groups, but just for now, let's talk about the types of symmetry. So the first type of symmetry is bilateral. That is what we are. It means we can technically be sliced from, please don't ever say that in a test, you will get it wrong. But our left side, our left hemisphere, and our right hemisphere are symmetrical to each other. Okay, if we drew an imaginary line down our midline from head to toe, we would find that the left side and the right side are mirror images of one another. That makes it bilaterally symmetrical. Okay, the next type of symmetry is called radial symmetry, and I want you to think, when you think of radial symmetry, of a nice piece of, well, not piece, a whole pizza. Okay, most of us have not had, well, all of us have not had access to Romans and debonaires and all the lovely pizza places. I hope I'm making you really hungry. And depending on how bored you've been on lockdown, some of us have made our own pizza and then failed miserably at it being symmetrical. But I want you to think of the perfect little Romans pizza. It comes nice and round in a box. And when they've cut it, they cut it into triangles that are all equal in size. For the most part, they obviously don't measure. But basically, it means that if we take the midline or the middle of the circle, think of, again, just keep with your pizza there. If you measured the radius and you found the middle of the circle, if you cut out a circle and you folded the circle for two halves to meet each other, it doesn't matter where you fold it, as long as you fold it through the middle, you're going to be able to keep folding it, right? I hope so. If you didn't fold it through the middle, obviously then it's not symmetrical. But as long as you had pinpointed that little middle centerpiece and you try to fold it in whichever direction, you would be able to fold it. That is radial symmetry. So it's symmetrical along many axes of the body. And then the last type of symmetry is asymmetrical. It means there is no symmetry. Doesn't matter how you try. If you find a midline and you try to find a mirror image, you're not going to find one. It does not have any similarities on one side or one axis as the other. Okay, so 3 types: bilaterally symmetrical, radially symmetrical, and asymmetrical, not symmetrical. Okay. All right, so I've said that. All right, and I've said that, so you guys can read through that yourself. Okay, and just to add a few more notes. If I talk about your brain and spinal cord, I can call that your central nervous system. Okay, it's the system that is going to be responsible for sending messages to your whole entire body. And then just a little bit of directional words again. If we talk about the anterior part of something, of an organism, anterior means the front or head region. Okay, if you as a human, your head is on the top, so anterior would be your front, okay, not your head only. If you think of a worm, the head would be anterior. If you think of a dog, the head is anterior, okay. Posterior would mean back or anal region. So if you think of a human being again, the entire back part that we cannot see of ourselves, that we look at only on other people, that is posterior. But if you think of a dog or a worm, the anal region, the bum region is the posterior region. And then if we talk about dorsal, we're talking about the top of an organism. So think of a shark and think of its dorsal fin. The top part of the shark is dorsal. And then think of then ventral means at the belly or at the stomach or the underside of the organism. All right. Okay, so that was symmetry. Just to go back, that was symmetry. 3 types of symmetry. We can ask those. We can give you diagrams and ask you to know the symmetry, but also the 6 phylums that we will be doing, you will have to know the symmetry of each phyla. But I could also, I don't know, this picture of the horse that you can see, I could also use something like this to ask you symmetry. I could give you any type of organism and ask you to find the symmetry for me or tell me the symmetry. Okay, so the next body plan is called cephalization. Now, cephalization just means that the sensory organs, the organs that are used to sense, so think of your senses, which are your sight and your hearing and your smell and your taste, and then obviously touch. But the majority of the sense organs are found at a definite head, okay, or anterior region. So in other words, if you think of humans, for example, we are cephalized organisms because our senses or our sensory organs, the organs that are responsible for our senses, are all found within our head. And then you're going to tell me, but ma'am, touch is our fingers. But if we didn't have a brain to interpret what our fingers feel, then we wouldn't interpret touch. And actually touch, you can feel touch everywhere. You can feel it if you brush up against something. You can feel your pillow on your head on the pillow. You can feel your toes in the sand. So touch is actually controlled by your brain. So all your sensory organs are all found in your head. And any organism that has cephalization has the same characteristic, which then tells us, grade 11s, that if an organism does not present cephalization, then their sensory organs are not found in one region, one anterior region. Okay, it is mostly bilaterally symmetrical organisms that display cephalization. In other words, organisms that have one axis of symmetry along a midpoint that have cephalization. Now, that cephalization allows the organism to respond or to move in a forward direction to respond to stimulus. Okay, so that means if I walk into Spa and I smell something nice and I think, wow, whatever that is, I want some of it. I can follow that smell, right? If I see my friend walking up to me, I can walk forward and give my friend a hug. If I hear someone calling me, I can run up to them and give them a hug. Or I can, if I'm lazy, I can stand where I am, but that's not the point. It also, though, allows us to detect danger. So I'm walking, let's say, I'm going to use Spa again. Maybe I miss Spa, I don't know. I walk into Spa and I spot that at the 6th till there is a guy who's holding up the teller, okay? And I realize, hold on, something's not kosher in the shop, I need to get out. I can immediately back away because I've seen the danger. So an organism that doesn't have cephalization would have a larger struggle with detection of danger and interpreting that danger for movement. Okay, so I know my Spa example is dumb, but if I'm an organism in the wild, it would be great to know that there's danger. Okay, if I'm just a tiny little squirrel and there's a, I don't know, wolf who's going to tear me to shreds, it would be good to see that there's a wolf so that I can climb the tree and get to the top. If I had no cephalization, I couldn't detect that danger immediately, and I couldn't respond immediately, or I couldn't respond to get away. Okay, so that is cephalization. The third body plan is how many tissue layers the embryo has. We call them germ layers. So again, I'm just going to remind you what I said earlier. When an egg and a sperm fertilize, they form a zygote. That zygote then starts to grow into a ball of cells by mitosis. And when it grows into a ball of cells, that ball of cells is called a blastula. That blastula, so let's say we have this ball of cells becomes 4 and then 8 and then etc., etc., etc. That ball of cells that keeps dividing and then specializes is called a gastrula. Now, a gastrula can have up to 3 layers, but sometimes it only has 2 layers. Now, that means that it has an outermost layer, a middle layer, and an innermost layer. Okay, so just providing some terminology there. We have ectoderm on the outside, mesoderm in the middle, and endoderm on the inside. So having said that, if you have a look at this, the ectoderm layer, the outermost layer, is the layer of cells that will differentiate into the skin, the brain, and the nervous system. Okay, so it does kind of make sense that it's all the outer things. The mesoderm, however you like to say that, will differentiate into muscle and bone and blood and skin and reproductive organs, which is your middlemost organs. And then the endoderm will differentiate into your digestive system and your respiratory structures, such as your lungs, which are the innermost, or which is one of the innermost organs that you possess. Okay, so when we look at those layers, as I said, an organism can have either 2 of those or all 3 of those layers. We then give them, each organism, a name based on how many of the layers it has. So an organism that only has 2 layers will be called diploblastic. Remember, di means 2. So diploblastic says it's got 2 embryonic layers, and it can't be any 2, okay? If it's 2 layers, it'll be the ectoderm and the endoderm, no middle layer. That makes sense, right? If you are 3 siblings at home, if you are part of 3 siblings, so in other words, you have 2 siblings, then you are 3 children at home, then you will have a middlemost sibling, a child in the middle. But if there are only 2 of you at home, then your parents can't exactly talk about their middle child. No one is their middle child. So if we have 2 layers, we can't have a middle layer. So we only have the ectoderm and the endoderm. And any organism that has only those 2 layers is called diploblastic. Right, then we get triploblastic organisms. These are organisms that have all 3 layers, the ectoderm, the mesoderm, the endoderm. You can ignore the EG for now, obviously, because I haven't done the examples with you. But when you are studying this, it's nice to have examples there. Right. I think this is the 4th one now. I think. Then let's talk about next, because my brain's not working. The next body plan key feature is the number of openings that the gut has. Your gut, what is your gut? Okay, so take you back to grade 9. We'll be doing digestive system later on this year, but take you back to grade 9 for now with your digestive system. I don't know if your teacher told you this, but this is how I teach it in grade 11. If you, let's say, for example, we had intestines that didn't turn and churn as much as they did. Basically, we are one long pipe from mouth to anus. Okay. It just has a couple of roller coasters in between. But if I cut you up, I did that Game of Thrones stuff again, and I spread you out over a football field, your digestive tract is one long tube from mouth to anus. Okay, so that is your gut. Now, when we talk about the gut of an organism, we can talk about a blind gut or a through gut. Now, a blind gut means that there's only one opening in the gut. Now, to put a nice pretty picture in your head, that means that it's not the same as the digestive system that you have. You eat with one end and you poop with the other end. If you had a blind gut, you wouldn't do that. And organisms with a blind gut, they ingest food and egest waste with the same opening. All right. If I talk about organisms then like us, who are mouth to anus, then I talk about organisms with a through gut. In other words, they have a mouth, one opening, and an anus, another opening. Some organisms have a cloaca instead of a anus, so for example, chickens. Okay. And you can see in this diagram over here, this yellow one, we've got mouth and we've got anus, and it becomes a through gut. Okay. That's the difference between a blind gut and a through gut, and we use blind gut and through gut to classify organisms. There are additional things, so you can have animals with a through gut that can be either protostomes or deuterostomes. Okay, we want you to know the terms, but we don't, because we only do 6 phyla out of about 30 phyla. We don't use protostomes and deuterostomes in our grade 11 classification, but it is used in classification. Okay, but we do need you to know the terms. So if I'm a protostome, it means that when I was just a little embryo dividing into a blastula, dividing into a gastrula, and my ectoderm, endoderm, and mesoderm were forming, it means that when my gut formed, the first opening, remember a blind gut, a through gut means I would have two openings. So when the first opening that grew, that appeared in the embryo, would become the mouth. Okay. If I was a deuterostome, it would mean that, again, I have a through gut, but when I was an embryo and when I was developing, the first opening that appeared was the anus, and the tube then developed and grew a mouth at the other end. Okay. So protostomes and deuterostomes just talks about which came first, the chicken or the egg, except it talks about which came first, the anus or the mouth. Okay. Right. The next key feature of body plans is called the coelom. Now, a coelom is a fluid-filled cavity. Right? It's a space that is filled with fluid, and it usually is formed within the mesoderm, so it splits the mesoderm. Okay. If there is a coelom, the fluid that is found inside that coelom is called coelomic fluid, and coelomic fluid acts as a hydrostatic skeleton. So if you think back, I think this was in grade 10, I haven't taught grade 10 for a bit, but you should have learned about jellyfish and hydrostatic skeletons, because you learned that they don't have bones, they don't have an exoskeleton, so they have a hydrostatic skeleton that is providing them with support. So animals or organisms either have this fluid-filled cavity or they do not have this fluid-filled cavity. And then there's some that fake it, but we'll talk about that just now. If an organism has a coelom, a fluid-filled cavity, it will split the mesoderm. Okay, guys, so you will see the middlemost layer will suddenly be split into two layers, and the coelom will be found in the middle of the mesoderm. Okay, that is a true coelom. A true coelom has to be found within the mesoderm, splitting the mesoderm. Okay, if an organism possesses that organism is called a coelomate organism. If an organism does not have that coelom, it is called acoelomate. Remember, a means without. Okay. The last one then, that's why I said some of them act, some of them pretend, they're good fakers. Some of them pretend, or they have something that appears to be a coelom, but it is found between the ectoderm and the mesoderm. I'm lying. It is found between the endoderm and the mesoderm instead of splitting the mesoderm. That's why I said it's very important to note that a coelom, a true coelom, splits the mesoderm. It's found in the middle of the mesoderm. Okay. If it is found between the endoderm and the mesoderm, then it is a pseudo-coelomate. Remember last year when you did animal tissues, you learned about pseudo-stratified. Again, there were cells that appeared to be stratified but weren't. Okay, so pseudo says appears to be, coelomate says coelom, so it says it appears to have a coelom, but it's not actually a true coelom. It's not a true fluid-filled cavity. So in this diagram, and I want you to ignore the little black dots because it's just the starting growth of the gut, so they all have that. I want you to look at the other layers. In this diagram over here, you see two layers, the purple and the blue, all right? So the light purple and the blue. So we have two layers. Innermost layer would be endoderm. Outermost layer would be the ectoderm. This then is a diploblastic organism with no coelom. So it's diploblastic acoelomate. The next one here, there are three layers, the light purple, the darker purple, and the blue. Three layers, but we didn't see a coelom, so it's a triploblastic acoelomate. Okay, and then this one down here, we see three layers, light purple, dark purple, blue, and then we also see within the dark purple, within the mesoderm, the middlemost layer, we find a cavity, this red part, which would then be a coelom. It's a true coelom because it's found splitting the mesoderm. So this would be a triploblastic coelomate organism. Okay, if this was a triploblastic pseudocoelomate, then this red would be found between the two purple shades, between the endoderm and the mesoderm. Okay. Right, so this is just a repeat again. Here you can see a coelomate, no coelom. Here you can see a pseudocoelom. Okay, you can see that where if you look here, this yellow layer is the endoderm. All right, and the pink layer is the mesoderm. So you can see that between the endoderm and the mesoderm, there's a layer. So that is a pseudocoelom. Here you can see the yellow and you can see the pink follows on the yellow, but inside the pink, inside the mesoderm, is a fluid-filled cavity. So therefore it is splitting the mesoderm and therefore it is a coelom. Okay, so this one is a triploblastic coelomate. This one is a triploblastic pseudocoelomate, and this one is a triploblastic acoelomate. Okay. Right. The next key feature is blood systems. Now, blood systems, you have either an open blood system or you have a closed blood system. The easier one to do, obviously, is a closed blood system because you'll recognize it. So you all know by this time, because you did it in grade 10, your heart sends blood away from the heart in an artery, which is then sent to your organs. The organs pick up that blood, use the oxygen in that blood, convert it to carbon dioxide, and send it back to the heart. Okay? So therefore, it's a closed blood system. The blood is never really leaving a vessel. Even when it enters the organ, it enters in a vessel. All right. Whereas with open blood systems, the blood does leave the blood vessel. So it will travel in a blood vessel and, for all intents and purposes, shower the organ that it was looking for with blood. So just think of literally, if you were a little organ, you would stand underneath the shower, switch it on, boom, be bathed in blood, receive the oxygen that you need, and that's the end of that. So there's no return system to the heart. So they either have an open blood system like that or a closed blood system, which I've then already done. Okay. So there we go. You can see that diagram. Right. Grade 11s, I'm going to end that video there then. Those are your key features of body plans. I'm just going to go right to the beginning so that you can have a look at them and we can compare them. There they are. Okay, so we've done symmetry. We've done cephalization. We've spoken about germ layers, which was the embryonic layers. We've done the number of openings in the gut. We've done the coelom. We've done blood systems. The thing that I've left out is the surface area to volume ratio. And the reason that I've left it out is because it doesn't get tested anymore. I will do maybe a short little video on it just so that it makes sense. But in previous years, they've done maths sums as SBAs for this, and I hate that. I think it's unfair as a question. But I will explain just the basics. In fact, let's explain the basics to you very quickly, and then I have covered that. Okay, so surface area to volume ratio says that the smaller an organism is, so surface area, right, would be smaller surface area. The volume the ratio of the volume inside that organism would depend on its size. So as an organism becomes bigger and bigger and bigger and bigger, there is a greater ratio between surface area and volume. Okay. And what I need you to know from that is just this little blue line. The smaller the cell, the more efficient the processes are. Guys, I want you to think about it. Okay. I don't know where you are at this point in time watching this video. I am in my bedroom with the door closed because I don't want an interrupting 3-year-old. So I will use the example of my bedroom. If I spray deodorant now within my bedroom, the smell, the particles will diffuse much quicker into this room than they will into the rest of the house. Okay? If I farted, for lack of a better example, the smell would be worse here than it would be in another room once I've opened the door. Okay. Now that's stupid, but the reason why I'm talking about particles is because of osmosis and diffusion. So let's leave osmosis because that's in water, but let's talk about diffusion. Okay. We know that diffusion happens in organisms, in cells, for example, for photosynthesis and cellular respiration to happen. The smaller an organism is, the better it's going to be able to do diffusion, and therefore it's going to have more effective processes. Okay, so a smaller organism would have better surface area to volume ratio and therefore more effective processes. This is what I was saying, that they sometimes ask us, they used to ask us to do the maths. I don't ever ask this. But basically, you can calculate a surface area to volume ratio like this. If I give you an organism, I can give you measurements. Okay, so let me do an example with you as it is here. If I tell you that something is 2 meters high, that is its length. No, okay, let's not do length, because I'm going to do height, so let's not do height. Let's do a ruler. Okay, a ruler is 30 centimeters long. Okay, everybody imagine your ruler. 30 centimeters long. Its breadth is, 3-4 millimeters. And its height is 6 centimeters, maybe, somewhere there. Okay, so that's just length and breadth and height to make you understand. Then let's give a better example just in terms of numbers. So let's say the length was 10 centimeters, the breadth was 2 centimeters, and the height was 4 centimeters. So I said 10 and 2 and 4, I think. Right, so the surface area would be length times breadth. That is 10 times 2, which is 20 centimeters. And area, so therefore must be squared. Volume is length times breadth times height. So 10 times 2 times 6. So 20 times 6, which is 180. And that would be centimeters cubed because it's volume. If I was going to work out the ratio, I would take the surface area divided by the volume. So 20 divided by 180. And if you just figure that out, 20 goes into 180 6 times. So it would be 1/6. Okay, that's the surface area to volume ratio. Okay, but so that's an easy one. And if anything, like I say, the previous government SBAs have been really tough that they've given on this, and I think it's unfair. But if anything, I could give you something simple like that in a test and say, okay, the length of it was this, the breadth of it was this, the height of it was this, calculate the surface area to volume ratio, and that is how you do it. So we're going to do surface area as length times breadth, and our answer is going to be in centimeters squared. Volume, we're going to do length times breadth times height, and our answer is going to be in centimeters cubed. And then we are going to take the surface area divided by the volume and find a ratio. If you answer that in 0, comma, blah, 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 you have not found a ratio. That is why I said if we have 20 over 180, then I think how many times can 20 go into 180? 6, great. So I've got 1 over 6. All right, that is a ratio. And as I said then, the most important thing is to understand is that the smaller the cell is, the better, the more efficient the processes are going to be. Okay, right, so up to there then, I am done with key features of body plans. What that means now, guys, is you can see in the slide it says 6 of about 30 phyla. We're only going to learn about 6 phyla, 6 groups of animals. I now say to you, because that's what's going to happen in the next lesson, I'm going to say this organism, this phylum, has cephalization. This organism, this phylum, is diploblastic. This phylum is acoelomate. Then you need to know what those things mean. And now you've done those definitions. So in other words, when I cover, by the time I cover the next section, which would be the characteristics of the 6 phyla, I hope that you've taken the time to go over this video and learned, if ma'am says cephalization, what the hell does that mean? Okay, so there you have it. This is a very quick section, so I just want to end off by saying, just to give you the reality of it, if we were at school doing this, we would learn this from Monday to Friday and be done. Okay, we would learn animal diversity in 4 to 5 lessons and be finished. Where, for example, in plant diversity, we took 3 weeks. Okay, so just to give you the sort of an idea of not how important it is, because it is very important and it's nice free marks. If those of you, if you don't really like studying, or if you don't like studying, but you don't understand a lot of bio, this is I'm lying. This is parrot fashion stuff. This is, you learn that it either has cephalization or it doesn't, and you know what cephalization is, and you know what a protostome is, and you know what a coelom is, and you regurgitate that information and you can get those marks. So although it's a small section, it is a nice marks section. All right. In terms of what I've just done with you, however, it's exactly like everything in bio. The chances of what I've just done being a big question is 0, because where your questions would come in rather is to know which organism is a coelomate, which organism is diploblastic or triploblastic. So that will come. But without being able to understand what it is, doesn't help you. Also, as I've said, I can ask you symmetry in an organism, or I can ask you to draw, for example, a triploblastic pseudocoelomate. I can ask that. And just by the way, if you were asked to draw that, you would literally draw circles, like in that one diagram with the purple and the blue. You wouldn't draw cells in between. You would just draw 3 layers, 3 circles for triploblastic or 2 circles for diploblastic. And if you're going to draw a coelom, you would just draw a cavity in the middle of the mesoderm. Okay, and then label it accordingly, obviously. Oh, sorry. Okay, that's me. Until next time, stay safe.